Okay, people, I think we can continue. Are there people out there still? Are there people still out there? Are they coming? Are they coming? Okay. So can you understand my English? More or less? Okay. So one thing that I would like to do, and maybe today is not the right day because we have this distribution of desks in the classroom, it would be to know about you. You know, where are you doing? What is your background, and so on. So, but maybe tomorrow we can, if we have a different distribution of the tables, we can make this kind of tour so you can introduce yourselves and share with the others where you come from, what are your interests, in what level of training you are, and all these things that are always interesting to have. Because, you know, I insist that I would like at the end to form a kind of team, not just to have the professor telling you stories, but rather having a team at the end of the process, okay? Okay, folks, I think that we can restart. We're still missing a couple of guys there. Will they come? All right, so synthetic biology is engineering, more or less, right? By application of engineering to biology. What is important in engineering, in mechanical engineering, electric engineering, and these things? So one important essential aspect of engineering is standardization, standards. One of the major criticisms of synthetic biology to biology engineers have to monitor and to measure physical, say, parameters, and also the way. So I think this is a very serious issue. So there is this concept that many use, and I don't know whether I will be offending anyone, saying that the only science is, and the rest is collecting stamps, right? And I'm a chemist myself, so so I don't want to offend anyone, but that there's a pinch of truth in this statement, because physics takes care of rigorous measurements, rigorous descriptions, interchange data, all these things, and in biology we don't have that. You know, it's very, very difficult. So the laboratory, in many cases, can be made in a different laboratory because the techniques are different, the units people use are different, this or that, okay? So in biology we have, for instance, this promoter is very strong. What do you mean by strong? Strong in respect to what, right? You know, this is 1 meter, this is 2 meters, this is 3 meters. You know what you are talking about. If you say a promoter is strong, to be frank, I have no idea what you are talking about. And so because in many cases promoter strengths are with fluorescent proteins as reporters, for instance, and then, you know, in your laboratory you use GFP, and the other laboratory you use another GFP, in the other place you use a yellow protein. In other cases they will use transcriptomics. In other cases they will use LAX as set as reporter. So in biology we have a serious problem with standards. And standards, in fact, are the very basis of modern engineering. And therefore, if we want to bring engineering to biology, we have to keep an eye on standards, standardization, and rigorous languages to describe. So in the history of engineering, there is a critical moment in year 1841. Believe this. To have a specific thread for school. You may say, what is, you know, right now we are so used to see a standard. You know, we have electric plugs, we have you know, everything is standardized, right? And we take for granted that things are standardized, but they are not. I mean, they are not unless you deliberately standardize things. So if synthetic biology, to be something close to, we have to be careful about standards, what can be standardized, what cannot be standardized, and this type of discussions. So just as a way of example, in this year, this gentleman, Whitworth, devised a standard for screw turns with a specific thread angle, thread depth, and a radius. And incredibly, before that, every industry used a different type of screws, and that's crazy. I mean, to us, this is completely crazy. But by that time, people use alone that in different, in Europe, in all this mosaic of countries in Europe, every country has a different measure. And you know why in Europe now we all use the metric system? Because Napoleon forced every country to use the metric system. So another case where history crossed with science. So excepting, obviously, the UK, that was never taken by Napoleon. I don't know whether it was good or bad, but in any case, so he made it to England, and therefore they kept the pounds and the yards and these things that are not the metric system. But in continental Europe, that was the part of occupied by Napoleon, we all had to take the metric system by force. It was forced to use. So, so during the 19th century, different standards were proposed, and in fact, the metro standard was one of the main products of the French Revolution. So, one of the decrees that the French revolutionaries proposed to the country was to get about all these private ways of measurement, and then to have a metro as the kind of unit, and to have a kilo, and you know that first definitions of kilos and metro were like standards that were fixed in the streets of Paris. They say, well, and there's various places in Paris. You go there. They say, well, this length is a metro, period, and we impose. Well, anyway, so before the advantage about the standards is that they allow to do amazing things that. So, well, this is one of the many examples of standards, but the value of standards is that they allow to uncouple and compare design from production, from assembly, from deployment. So in a standard or in traditional genetic engineering, one person has to do everything. Okay, you were given a project, you have to produce that can produce erythropoietin, or another hormone. You have to clone. You have to design the sequence. You have to produce the sequence. You have to clone it. You have to express it. By having standards, you can uncouple all the steps of the process as engineers do in the biological realm as well. So you can uncouple design from production, from assembly, from deployment. So in engineering, you can design a car in California. You can produce the parts, I don't know, in Kuala Lumpur or in Malaysia. You can assemble them in China, and then you can sell cars in Germany, And this happens because of standardization. Standardization, there will be no way this could happen. So by the same token, in biology, if we are able to develop standards, the number of applications that we can do with biology and biotechnology, right? So I just put this as an example. This is a city where I have some family roots in Spain. It's called Segovia, and it has the remains, or it conserves a very old Roman aqueduct. And I used to put this in as an example of standards because you know that this is a typical civil engineering endeavor. You have arches, you have stones, you have like specific distances between the different arches and so. So that meant that the engineer that constructed this was able to design that in a place the parts were produced in another place, and at the end they were assembled in another place. And this can be done only with standards. If you don't have standards, there is a lot of things that you can do. So, for instance, if you have a wood, you can take parts out of the tree and make a graft here. So that's fine. I mean, it serves some purpose, and there are many things that you can do there. Right? But you don't have a standard. You can put things together and hope that it works, right? But you have a concept. You have standards that can put together the planks and all the components of a ship that follow a given blueprint. Then you can make an object that can do much, much better than this other and can follow much longer distance. So, we, or humans, were able to cross the ocean. We were able to generate standards for ship construction. When you have, a big airplane, or way more complex. So, what type of standards? I will spend classes to standards and some efforts that are going on for standards. And one of the standards that is becoming very popular in synthetic biology is to describe bacterial promoters as logic gates. Coffee or tea. So a logic gate is a mathematical abstraction that, indicates this kind of small computational unit where you have an input and then you have an output that follow a deterministic logic, right? So I don't know how familiar with logic gates, but please, this is important to understand and to kind of grasp genetic circuits and this type of genetic constructs, so people can effort to remember the symbols that are associated with different logic gates. So there is this operator that is called an amplifier. An amplifier is that you have an input and the sign of the input is identical to the sign of the output. So if the input is 0, the output is 0. If it is 1, the other is 1. So these are digital logic gates, okay? So they are ones. Or, for instance, you can have a NOT gate. A NOT gate is an inverter. We mentioned that before. An inverter is that when you have a 0 input, the output is 1, and vice versa. So they are connected by a transfer function that is inverted one in respect to the other. So these are simple logic gates. But you can have more complex logic gates where what you have is 2 inputs and then 1 output. And one of the most, popular logic gates involve the AND gate. So AND gate means that to have a positive signal, you need the 2 inputs to be positive as well. So this is the so-called the table of truth. So you have A0, B0, the output is 0. If you have A0, you have B0. Only when you have A and B with 1 values, then you have a 1 value. So this is an abstraction. So how can we implement? These are some of the simplest logic detail other more complex logic gates and how to implement them with biological parts. So how can we implement this in the term implement is super important also in this narrative. So implement is to materialize, to embody a concept, put that in material terms, right? That's implementation. Okay, so for instance, an amplifier. An amplifier can be implemented through a promoter where you have the RNA polymerase activating the promoter only with the help of an activator, okay? So that means that when you have an activator, then the input is the activator, and then the promoter is on. If you don't have the activator, the promoter is off, because the polymerase will not bind there, okay? Also, to implement a NOT gate or an inverter, then what you have is the situation where you have a repressor that will block the entry of the polymerase in the binding site for promoter activation, okay? So that means that if the repressor is there, then the polymerase will not make it, and it will be 0. If the repressor is not here, then the polymerase will happily go into the promoter and activate transcription. Okay? Just a little detail for those who are not familiar with the terminology. This little bubble here indicates negation, negative. Okay? So you have an amplifier and you put here a little bubble. That means that the operation will now be negative in respect to what you had before. Okay? And you will see that there are other more complex gates where you have these bubbles all over the place. In an AND gate, how do you implement that? Well, you can also just keep in mind you can implement these gates not only with transcriptional factors and proteins and RNA polymerase, but with other tricks we'll share with you in some cases. So an AND gate, for instance, would be a situation where you have the RNA polymerase being activated by a transcriptional factor, but it happens that the transcriptional factor itself may be activated or requires to be activated by an effector. This is a typical case, for instance, and we will see some cases, a transcriptional factor that is inducible. Okay? So then to have this polymerase activating transcription, then or running transcription, you need to have not only the activator, but also the effector of the activator. You need A and B. Then the system goes in this direction. Okay? So then what happens is that you can make an abstract circuit here, and then implement the circuit by playing with repressors and activators here. We'll have tomorrow probably some specific discussion on what type of parts we can use to implement different types of logic gates in different configurations. So what is the advantage of using logic gates? Well, the advantage is that logic gates are composite, that the output of a logic gate can be used as the input of another gate. In such a way that, I mean, this is something that electric engineers know for a long, long time. In the case of biologists, what we do is to benefit from this type of concept, this type of nomenclature, to reshape the image of complex regulatory systems as they were a network of logic gates. What we have here, and you don't have to get the details, is a pathway for biodegradation of M-silane in a soil bacterium that is called Pseudomonas putida. So you have a number of inputs and you have a number of outputs. The final output, obviously, is production of the pathway for degradation of a given compound. And this slide here summarizes all the known interactions, whether positive or negative, between each of the components of this complex regulatory circuit. So at a distance, you know, you would say, well, this looks like an electric circuit, right? So you have connections and everything. You have bubbles here and there. You have NOT gates. You have, this is an OR gate. We will discuss OR gates in detail at some point. And so you have, so you see that by standardizing the transcriptional factors, whether negative or positive, in the shape of a logic gate, then you can make sense of the regulatory logic of some otherwise difficult to penetrate regulatory circuits, right? So that's one of the samples. I see that today I just touched upon various topics. We will get in depth into them and some other types. So there's another important aspect of standards, and it's what is called metrology. How do you measure things? So this is more physics, well, this is physics and engineering, but you know, how do we measure things in biology, right? So if you think about it, practically for many years, the only real unit of something in biology was enzymatic activity. Okay, so you know that you have enzymatic activity and you can measure, you know, transformation of A into B with these units per milligram. I mean, this is the only where, you know, or the only major case where you have a measurement of something. Or, for instance, fluorescence. But fluorescence is something that is not intrinsic. Depending on the reporter that you use or depending on the, say, material you use, fluorescence is not an absolute measurement. So, so how can we convert these kind of soft measurements into something that is useful for us to implement circuits with logic gates? Well, we ran into this issue of molecular metrology. So molecular metrology at the very, very basic level, has like two important parameters. One of them is transcription, and the other is translation. Well, you will see in the following classes that the basic fundamental core of synthetic biology is what is called the gene expression flow. So you have DNA, goes into RNA, and goes into proteins. So this is like the heart of the whole field of synthetic biology, because you have questions that you have to fill to convert that fundamental concept into an engineering tool. Okay, so because of that, you have transcription, and you have translation. Now, how do you measure, how you make some metrology of transcription? How can you produce an absolute measurement of transcription, okay? And instead of saying we have a strong promoter, a weak promoter, well, this promoter has activity. We don't use this vague term of weak promoter, strong promoter. We use a good term, as physicists do in their field. So years ago, someone called Drew Endy, that is one of synthetic biology, said, well, maybe we can say express absolute activity of RNA polymerase or promoter activity in something that we call POPs. So POPs means polymerase per second, okay? And these POPs would be the biological equivalent to electric current. I have 3 amperes, right? And amperes that goes given time through a given surface in a cable or in an electric device, right? So by this transcription, then that is POPs, so polymerase per second. So if you have many times that the polymerase goes through a promoter, you have a high POPs, and you can measure that. You can give a number, and if not, then you have a weak promoter. Okay, POPs, very important concept. Bacteria as a radio, one metaphor. Bacteria as computers, another. Okay, so the idea of bacteria as computers was put forward by the gentleman called Antoine Danchan in the Pasteur Institute. And well, you think what a cell does, is that comparable to what a computer does? Well, to some extent, yes, right? Because it takes information, it takes inputs, and then it releases and delivers some actions, some activities, right? So maybe this is something that can be incorporated into the metaphors of synthetic biology. So when you have these new metaphors, again, you can introduce new language, new methodology, and new agendas, as I mentioned before. Now, one basic aspect of computers that is super important for synthetic biology, and again, it's something that in many cases we don't think about it, is that one thing is the software and a different thing is the hardware. And they are completely separated things. The hardware and the software is completely different. So in computing or in computers, the hardware, is like the machine that you touch, that you can say materialize, and the hardware and the software is the program that you put there. You can put different types of programs, but you can use the same machine to exchange different programs. So by the same token, what if we consider bacteria and other biological systems as computers where they have a hardware, that is the gene expression flow machinery, but then they have a software, and is the program that we give the machine to interpret and to execute. With the real difference that in a bacterium, it is the same say hardware that makes copies of itself. You may say, is that unique of biological systems? I mean, you think that biological systems are the only ones able to make copies of themselves? 1, 2, 3? No answer? Okay. Okay, there is this super interesting project for many years that is called the REP. Can I write here? Okay, so it's called the RepRap project. And if you look in internet, you will probably find it. And it's a project that is about having 3D printers that print 3D printers, okay? And this is very interesting because, you know, at the end, it's exactly what we have in bacteria. So a printer that makes a printing that is a copy of itself, right? So that's another angle that I find interesting for this discussion on whether biological systems are completely different of the rest of the natural world, okay? So if you can make a machine that by itself makes a reproduction of itself, is that alive? Is that a biological system? No, it's still an engineered system. It's just that the program that it has enables making a copy of itself. So this makes, again, a parallelism between things that we observe in the biological realm and what we observe in the world of engineering. But again, returning to this, just keep in mind that bacteria, in many respects, can be compared or can be seen as, computers making copies of itself, computers that make computers. Okay, so what is the hardware of the gene expression flow? That is the machinery that is behind, that enables implementation of these circuits and enables that at the end the bacteria that we manipulate in the laboratory do what we want them to do. We will spend a lot of time discussing various steps of the gene expression flow because it's super important, okay? And it's quite complex, and this is something that perhaps engineers don't like very much, and well, but so be it. There's this famous sentence by Drew Endy about biology as compared to engineering, okay? And he says, I don't want an Airbus that goes in flight to develop emergent properties or to in the middle of the flight, you know? And this is something that, for better or the worse, we have in biological systems all the time. So we have a lot of complexity, and in every step of the complexity, something can go wrong. And this is something that synthetic biologists have to really pay attention to if you want to, at the end, have real, well-robust engineered systems. So this is something that is in all textbooks of biology, and is the famous so-called central dogma of molecular biology. By the way, again, if I have time, we have a little discussion on whether it is completely correct to assume that you go directly from DNA to protein and you cannot go back, but that's a different story. So for the time being, what we have is DNA, RNA, and proteins. But look, this is not a direct, event. It goes through various, stages, and in every stage, something happens that we have to control in order to have good systems for programming and for actual activity in whatever scenario we want it to work. Okay, so we have DNA and goes to RNA. More or less, this is a simple step, but then the fun starts after you have RNA. Because RNA is subject to various levels of stability, so it's not that you have a molecule of RNA and it's there forever. No, you have issues connected to RNA stability, and RNA stability may affect tremendously the outcome of a given gene expression project. Then on top of that, availability of RNAs for translation is subject to the action of other elements that are in the cells that are called small RNAs. So intuitively you think that all RNAs in a cell are long RNAs, but no, there are many, many small RNAs that interfere with the long RNAs and can really change entirely their activity and their profile. We also have this riboregulation, very important in synthetic biology as well, and it's the fact that the three-dimensional architecture of RNAs can change depending on various factors, including the interaction between RNA sequences and small molecules. And this is something that up front may look bad, but in fact, it has been used in some cases, and we will discuss that in a later class, for making devices that artificially regulate expression of given genes at the level of translation, not at the level of transcription. Okay, but then you may have translation at the end. But then again, the fun starts or restarts because there are many issues connected to protein stability, and also there are many issues connected to post-translational modifications. So as you see, this starts entering a challenge in our kind of agenda, because our agenda is to be able to program bacteria for given activities, and ideally, you write the program, DNA, and immediately you should have activity in the form of protein synthesis. But wait a minute. Between here and here, you have to deal with all these things plus others that I will discuss at some point. So what do you think is better? To make things simpler, or to try to understand how bacteria deal with the different steps and trying to leverage the different steps and the different mechanisms for our own sake? What would be your kind of approach? What do you prefer? To have something that is simple? I mean, you say, well, let's eliminate, for instance, iron stability, out. We take out a regulation by small RNAs, out. We take out protein stability, out. We take post-translational out. And then we have a system in which we go straight from DNA to protein synthesis. Okay, so there are like two schools in synthetic biology to deal with this problem. One of them is the school of what you may call the MIT. Well, one thing I have to tell you is that synthetic biology is something that is going on already. It's going on. It's not like it's not a textbook of physics that you know. You have all the definitions and everything for many years. It is something that is completely new and is completely being revised. And you have like different schools in the world that produce different opinions, you know. And some opinions are like this, and the others are entirely opposite. So what you may call the American branch, North American branch of MIT, of synthetic says, well, biology is very complex. What you have to do is to make it simpler. If you want to make engineering, you have a complex system. You have biology with all the little mechanisms, and so that biology is like very much, but they are a pain in the neck. So for an engineer, so what we have to do is to make things as simple as possible. So if you start with a situation where you have RNA, or you have DNA and you have RNA, and what you have to do is to make sure that you eliminate everything that bothers you, and you have simple things. So there's a company in the US that you may have heard. It's called Ginkgo Bioworks. Okay, so this company was created by some of the pioneers of synthetic biology in the US. If you have curiosity, you can go into the webpage, and the kind of logo of the company is making biology easier to engineer. So that's what they sell, okay? What they sell is, let's make biology easier to engineer. Okay, that would be the kind of American school. Let's make simpler, and the simpler they are, the better or more amenable to manipulation or to re-engineer there. There's the other school, to which I belong, say, and they say, well, instead of trying to fight nature and to kind of force nature to get a simplicity that might be difficult to obtain at the end, let's try to understand what is going on here, and maybe by understanding these steps, we may be able then to make a more robust, say, a flow in the gene expression flow. Which of the two approaches will work eventually? I don't know. I hope it's not them, but you know, who knows? And indeed, their approach is very successful in many cases, because obviously you say this is simple, that's more appealing for investors and this type of thing. You say this is complex, right? So you have to keep also this kind of PR aspect. Well, in any case, so that's going to be one aspect of discussion. So elaborating a little more with this idea of bacteria as computers, so we can really summarize the different types of actors in this scenario in this slide here. So what you have in the program, in the side of the program, is that you have a genome with all the DNA and so on, and then composition and then everything that you can determine genetically. And then you have a machine that once is able to interpret the program, then you can deliver a given phenotype, okay? So you have a chassis, and this is a word that will be the subject of an entire class because it's super important. So chassis would be, in the jargon of synthetic biology, like the basic structure of a cell that is basic functionality and enables also the possibility to insert in and out different devices that are also encoded genetically, okay? So the way at the level of the program this is translated at the level of the phenotype is that if you enter a genetic determinant into a genomic chassis, then the machine will interpret that as a new trait that will be then, say, expressed in a biochemical and structural fashion, and at the end, what you will observe will be a new function or a new metabolic trait, okay? So just keep in mind that this would be, in this slide here, what you have in the program is basically DNA. What obviously the molecule of DNA is not a program, it's the sequence that you have the DNA, what is a program, and then the machine will convert this DNA into something structural that will give rise to a specific behavior that you can monitor with your eyes, right? Okay, so that means, and this is another interesting and important angle of the whole discussion, that DNA and DNA synthesis and DNA production in large amounts in a deliberate fashion is perhaps the most important technology that has been brought up by synthetic biology. So encoding information of DNA might be complicated, but at the end, if a computer as encoder can kind of write a whole program with zeros and ones, then in biology you can write a very complex program with the 4 bases, right? So that means that if you want to program bacteria or different biological systems for a given purpose, the most important technology that you have to access to is DNA synthesis. If you don't have DNA synthesis, forget it. You better get another job. It's super important to have the possibility to write long sequences of DNA with a given program embedded in it. So this is something that in the past was super complicated. So I'll tell you a story of my life. So I worked in Berkeley in year 1986. And then just having a sequence of DNA of 20 nucleotides was a super big thing, You had to order it. Obviously, it was not commercial. You had to go to a guy that was working on this thing, and then it took like various weeks to have the whole thing synthesized. It was completely dirty. You have to purify it and everything. Now you call and you write a real message to a company, and then you have it next day, okay? So this is something that you have to see also the progress. But in any case, the ease and the facility now to synthesize long sequences of DNA is just amazing. And you know that right now, well, I don't know exactly, but at some point you have to tell me what your own experience. But right now there are many cases where cloning is not interesting any longer because you can really synthesize the genes directly. So once you have the sequence, then you order a company to synthesize the whole gene for you, and that's the end of your problems of cloning and these things. So DNA synthesis is one of the key tools for synthetic biology. Once you have the capacity to write a DNA, then you can just write a complex, sequence here. Not only this repressilator system that I mentioned before with an oscillatory behavior, you can write all you want. You can ask the bacterium to go to the right and then to go to the left, and then when it finds a given compound to emit light and then to commit suicide, for instance, and this is something that has been done. So, you can engineer behaviors because you can have access to DNA sequence that are not natural, but can be enabled by our ability to synthesize DNA. So this is one case, for instance, where I don't know exactly what is the outcome of this particular device, but you have a series of promoters, ribosomal binding sites, and genes, and they are encoded in a long DNA sequence, and they all recognize other members in the circuit. So at the end, you can generate, at the end, writing a program for a bacteria is not very different of writing a computer scientist to write a program, a code for a computer, right? Or for a given computer program. So this similarity between computing and, say, biological systems is also very useful as a metaphor, as an interpretative frame for this type of endeavors. So obviously, as I said, it's not, in this slide, I put future approach, where I have to say it's not future, it's already present. And once you know in silico what you want to do, what genes you want to put together, what promoters, what parts you want to put together, then you just order it to be done by a company. And right now there are many different ways of assembling long DNA sequences. And then at the end, you have the synthesized DNA in your mailbox, and the next day you go to the laboratory and use it with no problem. Well, with no problem or with some problem. Because what happens is that synthesis of long DNA sequences is always damped by errors. It's like when you write, when you type your letter in the computer, I'm sure that we all make mistakes, right? So the same happens with DNA synthesis. The machine can be programmed to synthesize long sequences of DNA, but it's nearly unavoidable that at some step there will be a mistake. And then the mistake will be, say, a fix in the DNA sequence, and then when you make your experiment, it may not work at all. We have in ourselves, and maybe some of you who work in this type of things, that you order a segment of DNA with a given sequence, you make your experiment, it doesn't work, you go back, and then you realize that the sequence of DNA that you ordered at the beginning was wrong. And therefore, having long sequences of DNA completely faithful to the program or to the initial sequence that you wanted to implement is not completely guaranteed. But again, this is something that is progressing, and maybe in the next few years, this will be a problem that will be solved. Okay, so this is, up to this point, like the general basis of synthetic biology. I insist that in the next few days we will see more in detail some specific aspects of what I told you today. And but I want to spend the last hour of this presentation discussing some examples on what can we do with all this. These are just a few examples because, as I told you, this is a field that is developing very quickly, and we are just at the beginning of the revolution that is being brought about by synthetic biology. But I'll show you a few examples that have also some historical value, in the history of synthetic biology. So one of them was this idea of programming, say, patterns in biology. Well, there are many different types of patterns. You have, in zebra horses, you have all these strips. You have patterns all over the place. Bacteria never form patterns, right? So there was this project that was led by someone called Ron Weiss in the MIT in the early 2000s that developed a circuit. I'm not going to get into the details, okay, because we are too early in the course to get into the details. That will come later. But what he did was to take different parts of his old bacteria, and he played with a phenomenon that is called quorum sensing. That I will explain in detail. But basically, the idea is that some bacteria produce some diffusible small molecules that are just released into the environment. And if these molecules find a neighbor, then that signal is converted into transcriptional activator of a target gene there, okay? And under natural conditions, this system is used for quorum sensing. So quorum sensing is that you are confined here in a kind of closed place, and then the system detects whether you have one member or two members or have a crowd around you. And because all members of the community are releasing this compound, so the moment that the level of this so-called autoinducer compound goes beyond a threshold, then that means that we have a bunch of people in the room. So by the same token, once you have a sufficiently high concentration of quorum sensing molecules, then the bacteria know that it's surrounded by other, say, partners